Okay, people, so we will discuss. See, we have already discussed how the factors are involved in the formation of clotting, how the different 12 or 13 factors, however we take it, are involved in the clotting of the blood. So the clotting of the blood is a very important and necessary step in order to ensure that there is no blood loss or there is no heavy blood loss from the injury site. Okay, and clotting of blood doesn't always mean that there is a injury. For example, when we are bitten by a mosquito, even then there is a blood clotting. Even when when there is an injection given to us, then also there is a blood clotting. Okay, so blood clotting happens not only at injury sites but also for various other things as well, and it is very important as well. So it's a very complex network and a cascade mechanism that we have seen. This is just the gist of it. Okay, what we just need to remember. Okay, at the place of injury, this is the minimum thing that we need to remember. Of course, the involvement of the 12 or 13 odd factors in the final production of fibrin and the mesh or the network in which the WBCs and the RBCs get trapped so that the blood is not oozing out, that has to be remembered as well. But this is the minimum requirement that you need to have as a knowledge base for the coagulation of blood. So coagulation of blood at the place of injury, when the blood vessels are damaged, so what happens, there is release of thromboplastin, and this basically triggers a chain of reaction. So then there is formation of thrombin, then there is formation of fibrin, and then there is formation of blood clot. So all these are basically chain reactions. So the blood taken out from blood vessels can be, now many a times we take blood out from blood vessels for pathological testing, or blood is also kept in blood banks to be administered to people who need it. Okay, so see, the moment blood gets exposed to air, the thromboplastin and all will start forming, right? Or the moment the blood comes in contact with there are two things: blood gets exposed to air, or it comes in contact with collagen fibers in the tunica externa. So under these two conditions, the blood is going to initiate a chain of reactions which is going to form clotting. So how to prevent clotting in the blood which is kept in blood banks or pathological treatments or for testing and all? So when the blood is taken out from the blood vessels, it can be prevented from coagulation by adding a pinch of sodium or potassium oxalate. So sodium or potassium oxalate, it will prevent. So since oxalate precipitates calcium ion, so we know that sodium and potassium are basically alkali metals, and calcium is alkaline. So obviously sodium and potassium will displace calcium ions from the blood. Calcium ion plays a very important role. It is factor 4 basically, and plays a very important role in the coagulation of blood. So the sodium or potassium ion oxalate, they precipitate calcium ions. So now the blood will not have any effect of calcium ions. So blood does not clot. So blood takes longer to coagulate at a very low temperature. So it will take very longer to coagulate at very low temperatures. Okay. Now let's say this is the wound site. So what happens is, at the wound site there is an injured tissue. The injured tissue releases thromboplastin. So blood platelets, the ruptured platelets, injured tissue, the thromboplastin is formed. Then we have the plasma, which has calcium ions, which has the prothrombin and which has the fibrinogen. So this prothrombin will convert to thrombin, fibrinogen will convert to fibrin. So if you look at the chain of reactions, thromboplastin in the presence of calcium ions or factor 4 will act on prothrombin to form thrombin. Again thrombin will act on fibrinogen, which is again a protein present in plasma, to form fibrin. Okay, and finally the RBCs and the WBCs will basically get trapped in this fibrin or long thread. Fibrin is basically a polymer. So the monomers, the fibrin threads basically are polymers, and they form a mesh or network of cells in which the WBCs and the RBCs get trapped, and hence the blood clotting happens. Okay, so this is the entire process of blood clotting. Okay, so just as clotting is very essential for the blood loss to be prevented from an injury site, so Anticlotting is also very essential, because if the blood is flowing normally in our blood vessels and clotting starts to happen, then the entire flow will be stopped, and that can be fatal to our health. Okay, so there has to be some anticoagulating agents constantly flowing in our blood to prevent the coagulation to happen. Okay, and that's something that we are going to study. Now, what are anticoagulants? So anticoagulants are substances which or basically conditions, to be very precise, these are substances or conditions. So anticoagulants are substances or conditions which stop the blood clotting process. Okay, and it is important as well, because under certain conditions, if blood clotting starts to happen, then it will create a different problem altogether. So there is one thing which is naturally present in our body, and that is heparin. So heparin is a natural anticoagulant found in our blood, synthesized by, and this is synthesized by mast cells. So mast cells basically synthesize heparin, and this is a natural anticoagulant that is formed in our blood. Okay, now we have heteropolysaccharide. Heteropolysaccharide basically, so this heparin basically is a kind of a heteropolysaccharide. This is found naturally in our blood. We will see some external agents as well, but this is found naturally in our blood. It is heparin. Heparin is basically a heteropolysaccharide. It increases the effectiveness of a substance called antithrombin 3. Okay, so it increases the effectiveness of this substance called antithrombin 3. If you remember, thrombin is something which is produced, thromboplastin, thrombin, and so on and so forth. We are not going to do the entire cascade mechanism, but thrombin is essential for the formation of blood clotting. So it produces antithrombin 3, which goes against the production of thrombin, and hence it is called antithrombin 3. So it becomes more active and will not let thrombin formation takes place. Okay? So this will not allow the thrombin formation to take place. Thrombin formation is one of the initial steps for the coagulation of blood. So it will not let the thrombin formation to take place. Then we will talk about some external. This is basically present in the blood. So heparin is a protein which is basically a heteropolysaccharide, and it increases the formation of a substance called antithrombin 3, which will not allow the thrombin formation to take place during natural flow of blood. Okay, now we have another substance called hirutin. Okay, understand there is an organism called leech, and these leeches are basically found in waters, and if you have heard, many farmers get often bitten by them. They suck the blood of farmers. So understand, when they are sucking the blood of these people, of animals or maybe humans, so obviously there is an injury site from where they are sucking the blood. So why is the blood clotting not happening at that place? The reason why blood clotting is not happening at that place is because the saliva of leech contains a protein called hirutin, and this hirutin, when it mixes with the blood, it doesn't allow the blood to coagulate. So this is an external factor. Okay, why are we studying this? Because this hirutin, just as it is not allowing the saliva which is secreted from the saliva of the leech, is not allowing the clotting of the blood at the place where they are sucking the human's blood. So in the same way, this hirutin can be added to the blood samples which are taken for whatever purposes, for pathological testing or for administering blood, whatever. So this can be added. This lowers the clotting, and the blood samples which are taken for different types of analysis can be prevented from coagulation, because if it starts coagulating, because it is exposed to air, so obviously the entire cascade mechanism will start, because all the ingredients of the cascade mechanism are mostly present in the plasma, and this mechanism will start coagulating the blood. So it will become thicker and thicker. So instead of doing so, the hirutin is added so that the blood sample can be taken for analysis. Okay, there is another, this is basically animal based protein. So this is hirutin. There is another plant based protein which is known as warfarin. This warfarin basically inhibits the formation of factors. As we saw, all the 12 or 13 odd factors are required for the proper formation of clotting. So this inhibits, this prevents the formation of factors 2 and 7, factors 2 and 7 and 9 and 10 as well. Okay, and what it does is basically it lowers the effectiveness of potassium ions, and this is how warfarin will act. It is obtained from plants. So this also can be added to blood samples in order to prevent their clotting, and they can be analyzed for whatever purposes they have been taken. Okay, now we have chelating agents. Chelating agents basically do what? They are mostly made up of alkaline metals, so that they can precipitate the calcium ions, which is alkaline. Alkali metals like sodium or potassium can precipitate the calcium ions, displacement reaction, which we have studied in chemistry. So chelating agents can bind with metallic ions, and calcium ions are precipitated. Sodium oxalate, sodium citrate, then we have EDTA, which is basically ethylene diamine tetraacetic acid. All these perform the function of precipitating calcium ions. As we saw in the earlier slides, that calcium ion is factor 4 and a very essential constituent of the entire cascade mechanism wherein clotting happens. So this can be done. Now keeping the blood at low temperature is another option, because if the blood is kept at low temperatures, so wherever blood banks are there, they are kept at low temperatures. At low temperatures, all enzymatic activities and reactions can be stopped, and hence blood clotting can be prevented. So all enzymatic activities and reactions can be stopped, and hence blood clotting can be prevented. So this is how we can take care of the blood clotting part. Okay, certain terms that we need to be aware of. Let's say there is a term called serum. So in a blood clot, the removal of fibrin from the plasma leaves a colorless liquid which is known as serum. Okay, so if we remove the fibrin part, this will lead a colorless liquid known as serum. When a clot is allowed to stand for a while, it can be seen as a straw colored fluid forming an upper layer. So this straw colored fluid basically forms an upper layer when a clot is allowed to stand for a while. So sometimes it can be seen oozing out of a scab. So if we form a scab on a cut, it can be seen to be oozing out. The serum cannot clot, and hence it is of medical importance. The serum cannot clot, and hence medically it is very important. Okay, then we have thrombosis. So thrombosis, sometimes the endothelium of blood vessels is damaged or becomes rough. So the endothelium part becomes damaged or becomes rough. It causes the breakdown of platelets. So the platelets are basically broken down, which in turn forms a clot. This basically forms a clot. The patients are given heparin. So they are given basically heparin. As we see, heparin is an anti-clotting factor. Injections extracted from the liver of other mammals. So from the liver of other mammals, heparin is extracted and given as injections. Okay, we have seen that heparin is basically formed to ensure that blood clotting doesn't happen. Okay, so now we have hemophilia. It is basically a hereditary disorder due to genetic mutations, and when the blood does not clot, or even a very small cut, the blood does not clot, and from even a very small cut, heavy bleeding happens. Okay, so it is because the absence of an essential clotting factor, and this happens due to gene mutations. So these are certain things which we should be aware of. Okay, so now we will talk about the main pumping organ, which is heart. So we have discussed about blood, we have discussed about various components of blood like RBCs, WBCs and all. We have discussed how clotting takes place, so and how the gaseous exchange takes place in blood, and so on and so forth. Now what we need to discuss is how the entire pumping system takes place. So heart is basically the pumping organ, and in different places the structural and functional parts differ a little. Okay, so first we will see, whenever we try to understand an organ, we have to understand see, we will study the hearts of some other organisms as well. Primarily our study is on human heart, but a little bit of knowledge for other organisms is also required, because for any organ we need to understand the evolutionary aspect of it. Okay, so if the heart in a human is developed, obviously the other animals it is not so developed. So how has the evolution happened? What are the changes that we see in human heart? as compared to other organs organisms. So that's the reason a human heart has to be studied primarily, but in context with or in comparison with the heart of other organisms as well. Okay, so heart, basically we all know it will have chambers or compartments. So a heart is basically, see, we have, if I take the human heart, so these are basically auricles, these are basically auricles, and these are ventricles. So the heart enters its blood into the auricles and it passes into the ventricles. Okay, so the place where the blood actually enters is known as the auricle, and the place where it is pumped is known as the ventricle. Okay, so we have chambers, we have some accessory organs to these auricles and ventricles, but the chambers are formed from auricles and ventricles, and then we have some accessory organs which help in the functioning of auricles and ventricles. So there are chambers or compartments, atria or auricles. Okay, so as I said, atria or auricles, they are the receiving compartments. So the atria or the auricles are the receiving compartments of blood. For example, in human heart, through the superior and inferior vena cava, the blood, the deoxygenated blood enters the heart. So that is called the auricle. And from the lungs, the oxygenated blood enters this auricle. Okay, we will talk about left and right auricles and subsequently right and left ventricles as well. So there is oxygenated blood from the lungs which enters the auricles, and at the same time deoxygenated blood from the various organs of the body also enters the auricles. So auricles is basically the entry part. From the auricles, the blood passes on to the ventricles. So ventricles are the pumping compartments or chambers. So these are the ones which will pump and make sure the blood either reaches the lungs or it reaches the entire part of the body depending upon the organism. Okay, now there are two known as, so these are known as true chambers. These two, auricles and ventricles, these are known as the true chambers of the heart or main chambers. Auricles and ventricles together are known as the true chambers or main chambers of the heart, or they are also known as primary chambers. So these are the different names by which they are known. Okay, now these auricles and ventricles may be assisted by accessory compartments or structures. So these accessory compartments or structures, what are they? We might have sinus venosus, then we have conus arteriosus, and then we have truncus arteriosus, and then we have bulbus arteriosus. Okay, so these are basically helping organs: sinus venosus, conus arteriosus, truncus arteriosus, and bulbus arteriosus. So let's take an example of the heart of fishes. Heart of fishes. So how will it look like? So the heart of fishes has two primary auricles and ventricles. There are two auricles and ventricles, and there are two accessory organs. Two accessory organs: sinus venosus and conus arteriosus. So the primary structures, that is the primary chambers are there, that is auricles and ventricles, both are there, and we have sinus venosus and conus arteriosus. So how it looked like? This is basically the sinus venosus. This sinus venosus is basically a portion where the blood enters, and this is basically deoxygenated blood coming in from the various chambers or coming in from the various organs. So organs have used the oxygen, we will see where the oxygen comes from, but the organs have used the oxygen, so obviously the deoxygenated blood is coming and collecting in the sinus venosus. So as we said, sinus venosus is an accessory organ. It doesn't come directly to the auricles, it comes to the collection center of sinus venosus. From the sinus venosus, it basically enters the auricles of the fishes. Okay, now from the auricles, so we still know that this is deoxygenated blood which has entered, this is also deoxygenated blood which has entered. From the this now enters the ventricles. Again, this is also deoxygenated blood. Okay, so this is also deoxygenated blood. So from the auricles it enters the ventricles, and from the ventricles it is pumped not into the aorta but conus arteriosus. So before getting into the aorta will start somewhere from here. Okay, but before it gets into the aorta, it passes through the conus arteriosus. Then it moves into the main aorta. So the question is that what is the purpose of all this? Okay, because entry is deoxygenated, exit is deoxygenated. So what is the heart doing? Okay, so the heart is basically pumping the deoxygenated blood. Yes, you heard it correct. Deoxygenated blood. Why? See, in humans, the blood which is pumped into the aorta is not deoxygenated, it is oxygenated. But in fishes, the blood which is pumped into the aorta is deoxygenated, which means that where will this oxygenation happens? So the heart is just pumping, receiving deoxygenated and pumping out deoxygenated blood as well. So this will move into the gills where the gaseous exchange takes place, and that's where the deoxygenated blood will convert into oxygenated blood. And again, so here the pumping organ heart is just circulating the blood. It is not taking care of any oxygenated or deoxygenated part. Okay, the gills are going to take care of that, and that's how the hearts of the fishes work. So heart is only receiving deoxygenated blood. Okay, now this is known as branchial heart or venomous heart. Venomous heart, why? Because it is just receiving deoxygenated blood. Our heart is not venomous heart. Why? Because venous heart, sorry, because it is receiving oxygenated blood as well from the lungs. Okay, this is the reason why it is known as single circulation path. See, understand this. In humans, blood enters the heart twice. Once the deoxygenated blood, this deoxygenated blood is pumped into the lungs, and again the oxygenated blood from the lungs enters the heart. So that's the reason it is called double circulatory system. In fishes, it's only the deoxygenated blood which enters, and it's only the deoxygenated blood which exits, and hence it is known as single circulatory system. Okay, so we have here, so from the sinus venosus, it enters the auricles, and from the auricles it passes on to the ventricles. From the ventricles it passes on to the conus arteriosus, and from the conus arteriosus, this is basically the cycle: sinus venosus, auricles, ventricles, conus arteriosus, and then it passes to the aorta. From the aorta it passes on to the gills. These gills are the places or the site where the gaseous exchange takes place, and then it passes on to the other organs. Then it passes on to basically the organs where it gets deoxygenated. So now the gills have converted the deoxygenated blood into oxygenated blood. This oxygenated blood passes on to the different organs where it is obviously used up. The oxygen is used up, so it is rendered deoxygenated. This deoxygenated blood again comes back to this sinus venosus. So this is how the cycle is completed. So if you see, the blood enters the heart for the purpose of pumping only and not for purification. So in our case, the blood will enter the heart, and the heart itself is the one which pumps into this. So the same blood which is pumped, which is entered the heart, is pumped into the lungs, and from the lungs again it comes back. So that's the reason the same volume of blood is coming in and again going to the lungs and again coming in. So that's the reason ours is a double circulatory system, whereas in fishes it is a single circulatory system. That blood is deoxygenated, it enters while it enters, and it is deoxygenated while it leaves as well. It gets oxygenated only at the site of gills. Okay, so this is the difference between single circulatory system, which is fishes, and the double circulatory system, which is generally seen in mammals. Okay, so we will talk about different aspects of heart as well. I don't know whether this is visible. Let me just check if this diagram is visible. One second here, let me make some arrangements. Okay, so we are now talking about heart of vertebrates. Okay, now when we talk about heart of vertebrates, we have to basically recognize the progressive evolutionary changes that have happened in the heart. Okay, so in structure and function of heart, what has actually happened? So in vertebrates, basically their heart is single chambered, but if you talk about vertebrates, their heart is multi-chambered. So in invertebrates we have a single chambered heart, whereas in vertebrates we have a multi-chambered heart. So we talked about fishes, which are the simplest form of vertebrates, and we saw how in fishes they just have two chambers, auricles and ventricles, and they have some accessory organs of sinus venosus and conus arteriosus. Okay, so again the system which is there in fishes is single circulatory system, which means that the blood enters only once through the system. Okay, now mammals are the most evolved and have the most efficient heart. Okay, so we will start with amphibians first. Okay, now amphibians are basically a three chambered heart. So they have two auricles, that is the right auricle and the left auricle, the right auricle and the left auricle, and they have one ventricle. So the heart would, I guess that part is missing, so I will make a diagram here only. So what happens is we have here a right auricle, and this basically enters. So this is how we do the labeling part. So we have to be a little precise about the labeling part here. So how we label is, we stand like this. This is our left part, so we will label this as left auricle. This obviously is the right auricle, and this is the single ventricle that we have in case of amphibians. Okay, now there is, as we saw in fishes we had sinus venosus, right? We had sinus venosus. Now this sinus venosus used to collect the blood before pushing it to the heart. So in this case also the blood gets collected in the sinus venosus. So we have the, I guess this is not visible, so I'm just making it here. So this is basically the sinus venosus part, wherein the blood gets collected and it is pushed into the ventricle. Now from the ventricle the blood actually passes into the lungs, and we have this is basically truncus. So what has happened here is, in the accessory organs we do have sinus venosus as it was there in the case of fishes. Along with it we also have the conus arteriosus, but the conus arteriosus has actually fused, this conus arteriosus has fused with the aorta, and this has formed the truncus arteriosus. This has formed the truncus arteriosus. So in case of amphibians, the difference is that conus arteriosus, which was basically the tube like structure which actually passed on the blood to the aorta, that has modified by combination with aorta to truncus arteriosus. One more difference that is there, in case of fishes we saw the blood entering like this and passing up. So in case of fishes it is a tubular structure. In case of fishes, how it was a tubular structure. So this is how the blood used to enter, sinus venosus, and then from the auricle, this is the auricle, and then it passes into the ventricle. So this is the tubular structure that was there in fishes. Now in amphibians what has happened is, ventricles is on the lower side, and auricles is on the upper side. So this is another difference that we have in case of amphibians and fishes. Okay, so now so we have basically auricles are partitioned by a septum, and right auricle receives the deoxygenated blood. So auricles are partitioned by a septum. So there is basically a septum out here which partitions so this receives the deoxygenated blood, while the left auricle receives oxygenated blood. From the lungs it receives oxygenated blood. So the auricles don't mix up blood, but in the ventricle part the blood mixes. Okay, so ventricle is not divided and receives both oxygenated and deoxygenated blood, and hence the heart pumps mixed blood to the organs. This process of pumping in mixed blood to organs is known as arteriovenomous heart. The heart here will be known as arteriovenomous heart. So the mixing of oxygenated blood and deoxygenated blood is prevented by conus arteriosus. This is basically conus arteriosus on the aorta, which basically forms the truncus arteriosus in the auricles. But ventricles, there will be mixing of blood. Okay, so this portion is known as arteriovenomous heart. So arteriovenous heart is basically a heart which pumps both oxygenated and deoxygenated blood to the organs, because there is no septum, or there is no proper division between the ventricles. So the sinus venosus divides the conus arteriosus in such a way that oxygenated blood is passed through the body and deoxygenated blood is passed into the lungs. So the sinus venosus divides the conus arteriosus, which is basically modified into truncus arteriosus, divides it in such a way that oxygenated blood is passed through the body and deoxygenated blood to the lungs. So this system, as we saw in fishes, it's a single circulatory system. Why? Because the blood which is pumped into the organs from the heart passes through the heart only once. But here the blood which is pumped into the organs passes through the heart twice. Once it comes here as deoxygenated blood, it is pumped into the lungs, and again from the lungs the oxygenated blood enters here. The same blood coming in through different organs into the heart collects in the sinus venosus and is pumped into the auricles. Through the auricles it gets into the ventricles pump it into the heart. From the heart the oxygenated same blood is just a difference of deoxygenated and deoxygenated. So the same oxygenated blood enters the ventricles and again it is pumped. So that's how it enters and that's how it is collected. Okay, so this is called a double circulatory system. So what have we noticed as the primary differences between fishes and amphibians is that fishes, it is only two chambered, that is one auricle and one ventricle, whereas in amphibians we have three chambered, which is basically a left auricle, left ventricle, right auricle, left auricle and a ventricle as well. In fishes the auricle is on the lower side while the ventricle is on the upper side. The blood enters the auricle though, and but this is how it is represented. But in amphibians the ventricles are on the lower side, the ventricle is on the lower side, the auricles are on the upper side. Sinus venosus is similar as we have in fishes. So in amphibians what undergoes a change is basically the conus arteriosus. The conus arteriosus which basically carries blood into the aorta in the fishes, that along with the aorta merges with the aorta and changes, the fusion changes it into truncus arteriosus. So that's one difference. The other difference is that in fishes it is only deoxygenated blood entering and deoxygenated blood going out. Okay, and this basically passes on to the gills where it gets oxygenated, whereas in amphibians there is mixing of blood that happens in the ventricles. The ventricles are not separated by any kind of septum as we have in case of auricles. So in auricles the oxygenated and the deoxygenated blood do not mix, but in ventricles they mix. So this mixed blood is supplied to various organs of the body, and this is known as arteriovenous heart. So arteriovenous heart basically means mixed blood being supplied to all parts of the body. Okay, so this is a double circulatory system because the blood enters twice, and there are some modifications for the accessory organs, but basically the conus arteriosus changes into truncus arteriosus, and from single circulatory system the changes have happened to a double circulatory system. So that's basically the evolutionary pattern that has happened. So we will learn more into this. We are yet to do with reptiles.